Hello, everyone. We're going to start chapter 10 here. This is on meiosis and sexual reproduction. This is the end of unit 4, and it's a chapter after mitosis. Now, the great thing about meiosis is that it's very similar to mitosis. So, a lot of the same concepts that you learned in mitosis are going to apply to this chapter. With that being said, let's get into it. Meiosis is the type of cell division where the chromosomes are going to be halved and you're going to produce sex cells. Now, in mitosis, you produced body cells or somatic cells. In meiosis, you're producing sex cells. So that's one of the big differences between mitosis and meiosis. With mitosis, you're going to go from, in humans, 46 chromosomes in a cell to also having 46 chromosomes in each new cell. In meiosis, like I said, you're having the number of chromosomes, so you go from 46 to 23. And this should sort of make sense because, again, remember, we're creating sex cells, and your sex cells should have half the number of chromosomes compared with the somatic cell number. In humans, like we were talking about, the diploid number is 46, the haploid number is 23. Another important thing to remember here in meiosis is that we're creating genetically different cells. This is because we need genetic diversity in a population, and this is going to help with evolution. But with mitosis, the end cells are going to be genetically identical. So that's the main difference between meiosis and mitosis. Let's just kind of review a couple terms here that we need to know. Diploid, this is the total number of chromosomes within a somatic cell. These consist of the diploid number, or the homologous pairs of chromosomes. The haploid number is the half the number of chromosomes, and there is only one copy of each chromosome here. So they're non-homologous. You can see here they are different. You can see the homologous versus non-homologous. And this is just a reminder again, you get half of your chromosomes from your mom, half your chromosomes from your dad, and you get specifically one of each homologous pair of chromosomes from each parent. So you can see here in this first pairing, you get one from mom and then one from dad. This is why we have 46 chromosomes in our cells. Homologous chromosomes that have the same length and centromere position. So you can see here, these are homologous because they are the same length and their centromeres are in the same location. Alleles, these are different versions of a gene. Homologous chromosomes will have the same genes, but they may have different alleles. The example here is the gene for eye color. You may get an allele for brown eyes or an allele for blue eyes. So the alleles here might be different, but they're in the same locations because they're in the homologous chromosomes. Here's another way of looking at it. I love this image here. You can see homologous chromosomes, they have the same genes, but they may have different alleles. Non-homologous chromosomes don't have the same genes and the same alleles, and the centromeres are in different locations. Sister chromatids will have the same genes and same alleles because they're attached at the centromere. You can see here they are the same genes and same alleles. Now, it's important to remember that the chromosomes are not condensed or for a long period of time. They're usually uncondensed. They don't condense until meiosis or mitosis. But in interphase, when the cell is going through normal cell division, it's going to be the DNA is going to look like this. They condense and they look like this. However, they're not organized until humans organize them, and now computers organize them for us. But this is called a karyotype. And we're going to come back to karyotypes later on in this chapter. But just looking at these karyotypes, you can see here, one's from dad, one's from mom, one's from dad, one's from mom. So these here are a pair of homologous chromosomes, but if we look at this specific chromosome and like this one, they're non-homologous. You have to be the same length and have to have the genes in the same location. All right, to end this section, 10.1, we're going to talk about autosomal versus sex chromosomes. Autosomal chromosomes, these are chromosomes that don't code or don't determine the sex of an individual. Humans have 22 pairs of these autosomal chromosomes, so these are 1 through 22. I'm going to go back here. If you look at 1 through 22 here, you can see that the chromosomes go from largest to smallest, and then our last pair are X and our Y. These are our sex chromosomes. They're just a pair of chromosomes that determine sex. XX is female, XY is male. Now, I like to add in here, there are a lot of factors and a lot of differences within like these sex chromosomes that we're going to talk about at the end of the chapter. But it's not just XX and XY. There's just a lot of factors and a lot of things that determine the sex of an individual. So it's not just plain and simple. There's a variation here. All right, that's the end of 10.1. All right, 10.2 is going to look at the phases of meiosis. And specifically, let me make my thing a little smaller. Okay, good. So There are two specific divisions in meiosis. If you remember, in mitosis, there was only one. Within meiosis, there are two divisions. You have meiosis 1 and meiosis 2. Normal interphase occurs before meiosis, so that's where replication is going to occur, but there's not going to be an interphase 2. We don't need to replicate the DNA because it's already going to be replicated in interphase 1. You'll see why this is the case later on. Sometimes we call the time between meiosis 1 and meiosis 2 interkinesis. I don't specifically go into that much detail, but it is sometimes known as interkinesis because it is cytokinesis, and then there's a resting period between the two phases or the two stages. Now, I'm just going to leave this up here for a second, but crossing over is just the exchange of genetic material during meiosis 1. I'm going to show you what that looks like here, but basically crossing over increases genetic diversity. And let's look at the diagram that goes along with crossing over. So you can see these are pairs of homologous chromosomes. They're going to flip-flop genetic information. They can just exchange genetic information. And what that does is it increases genetic diversity here. So each one of these chromatid is going to be a little bit different. So you can see here they flip-flop genetic information. So each chromatid has a different set of genetic code. Now, this image is only showing us crossing over occurring once in that leg. It can occur hundreds of times between these two chromosomes, and they will exchange genetic information between both of these chromosomes. So I like to show that crossing over, but understand this occurs much, much more than just one time per chromosome. But basically, this increases genetic diversity, and this occurs in prophase 1. In metaphase 1, we have something called independent assortment. And this is when the homologous pairs of chromosomes are going to separate randomly or independently of each other. And this looks specifically at the maternal versus paternal sides of the chromosomes. Again, this is really kind of hard to understand as you're writing it down. Let me show you what that looks like visually. So we have three sets of homologous pairs of chromosomes. You can see we have a set here, and a set here. Each one of these combinations is possible. You can see let's say the blue versus the red. We have blue all on one side, then blue, blue, red, red, blue, red, blue, blue, red, blue, red, red, blue, blue, red, red, and then all reds. Each one of these combinations is possible because they're randomly independently assorting from each other. So this blue on this side, it doesn't mean that there has to be another blue on this side. Every single combination here is possible, and there's only three sets of homologous pairs of chromosomes. So if you do the math, there's over 8 million combinations for our 23 pairs of chromosomes. Add in fertilization through the other side, so you have a sperm and an egg, there's over 70 trillion, and then add in one event of crossing over. What we're showing here is you're getting a lot of different genetic variation within these sex cells. And that should make sense because there's so much genetic variation within a population. All right, anaphase 1, the homologous chromosomes are going to pull apart. So you can see here, this is much different than mitosis because in mitosis, the chromatid split. Now in meiosis, the homologous chromosomes are going to be split. So it's not exactly the same, but it has some similarities. Like I said, interkinesis is a rest period between the meiosis 1 and meiosis 2. DNA replication does not occur because it already has occurred. So you see here, these chromosomes aren't going to replicate again because they can't. They're already duplicated chromosomes. At the beginning of meiosis 2, cells are haploid, so they have half the genetic information. If we go back here, if you look at the beginning cell, it had 4 chromosomes. Our cells now have 2, so that means they're haploid. They have half the number of chromosomes. The great thing about mitosis and meiosis 2 is that they're exactly the same with some just minor details. Cells are haploid instead of diploid, and the genetic code is different. If you see here, our cells are genetically different, so that's one big difference, and then cells are haploid. But basically, they are the same. You can see here the chromosomes lining up, chromatids splitting apart, telophase looking like 2 of the cells here starting to form, and then cytokinesis. All right, 10.3 is going to look at genetic variation. This is when meiosis produces genetically different cells compared to mitosis that create genetically identical cells. Now, the two locations where we're going to see genetic variation within meiosis is crossing over and independent assortment. Yes, you can get mutations to create that genetic variation, but we don't need those mutations to create genetic variation within meiosis. We can do that through crossing over, the exchange of genetic material, and independent assortment. So this is how we create genetic variation without even changing the DNA code. Now, genetic variation creates a better chance for a species to survive. This is a huge evolutionary advantage, and not every species takes advantage of having genetic variation. I say genetic variation, but meiosis will more specifically say. Like bacteria, they don't go through sexual reproduction, but they still have variation through mutations. Now, their variation isn't as much as something that goes through meiosis and sexual reproduction, but there is a trade-off there. They reproduce a lot quicker. Are creatures that have that go through sexual reproduction, they have more genetic variation, but it does take longer for them to reproduce. So there's a trade-off, but this is a huge evolutionary advantage. Now, I'm going to relate this back to the next section. I shouldn't say back, I should say forward to the next section or the next chapter. Meiosis. When we see this independent assortment, this is where we're going to see the genes independently assort from each other. And this is why we have to make these Punnett squares. There's a big connection with this chapter and the next chapter. What we're seeing here is the progression of these chromosomes to separate, and that progression allows the occurrence of any one of these types of genotypes, which we're going to talk about in the next chapter. So just understand, meiosis produces what we see in these Punnett squares. And I like using this illustration to show the inheritance of the chromosomes from your mom and dad. You inherit chromosomes from your grandfather and your grandmother that they passed on to your mom. And each one of these chromosomes has parts of each. So you can see here the dad's chromosome is parts from his grandparents or his parents, your grandparents. And you can see that you are 50% like each of your mom and dad, but you are 25% like your grandparents and 12.5% like your great grandparents, and so on and so forth. But that genetic variation comes from meiosis and sexual reproduction. Same here. You can see you are parts of your your grandfather and your grandmother, and your father and your mother, and you can see on both sides what that looks like. I love showing these because it helps students understand, hey, you are parts of your mother, but you're parts of your grandmother and your grandfather, and so on and so forth. Now, we're going to talk about, we'll come back to that later on when we're talking about the trisomies. Let's just end with the differences between meiosis and mitosis. Mitosis, there is one division. You create two diploid cells, and cells are genetically identical. This creates the somatic cells or your body cells. In meiosis, there are two divisions. You create four haploid cells, and cells are genetically different. You create germline cells or gametes, and that's the main difference between mitosis and meiosis. This is a very important slide. You have to know the differences between mitosis and meiosis. All right, 10.4 is going to specifically look at the life cycle. A little bit more in depth. Sexual reproduction. This is when two haploid cells are going to fuse together to form a diploid zygote. Obviously, these cells are like the sperm and the egg. That's as far as we really go with sexual reproduction. But these sex cells are known as gametes. They're just haploid forms of the sex cells. So thing to understand here is every species, this kind of works a little bit differently. So don't think like, how it works for humans and the sex cells. It's different for every species. There's a lot of variation here. It's not just as simple as sperm and egg sometimes. So there is variation here within this content, and that's species based. Zygote, this is the diploid form of the egg. You can see what that zygote looks like. That zygote will develop into something called a blastocyst, and that blastocyst will develop further on pregnancy. And that's honestly as far as we really need to go in this class. I like to remind students, remember, you're going to get 23 chromosomes from your dad, 23 chromosomes from your mom, and then this is the zygote. This is where you get 46 chromosomes. These egg cells and sperm cells are known as gametes, and they're haploid. They have half the genetic information or half the chromosomes. This baby cell is a diploid cell, and it's because it has 46 chromosomes, or it has the homologous pairs of chromosomes. Now, when we show you meiosis, we show you this process where the chromosomes split, and then we show these cells here. These are haploid cells. We don't show this secondary process. This is called spermatogenesis, and this is where these cells will develop into mature sperm cells, where they have a head where the chromosomes are, a body where the mitochondria are, and then the tails that propel them forward. So this occurs in the testes of guys, and this is just how the sperm cells are further produced after meiosis. Now, males will usually produce sperm throughout their whole life, but females, they go through a different process. This is known as oogenesis, and this is where the cell will split. And this actually doesn't produce 4 egg cells. This only produces 3 this produces 3 polar bodies, which are not egg cells, and 1 egg cell. You can see here, this is the egg cell that we're going to go for. This occurs in the ovaries of females. So it's a little bit different, and it's because they put a lot more nutrients and energy and just time and just, just energy into this one cell, because there's a lot more, how do I want to say, how would I want to say this? They have to devote more energy just throughout the pregnancy, so they devote more energy and time into this one egg. Guys, on the other hand, we produce a lot more sperm. We don't develop it. It's just how males and females have evolved over time. And I'm not going to go much more further into this. It can be complicated, but this process, again, is known as fertilization. I don't have you write it down because I think you should probably know this. But usually at this point in class, I'll show you a video. I'm not going to show it here, but it's a video just on sexual reproduction and how it occurs at the molecular level. And it's a pretty interesting one. It shows how sperm and egg interact, but this is where 10.4 stops. All right, 10.5 is going to look at karyotypes and how they are a visual display of the chromosomes. They're usually arranged by size, shape, and the banding pattern. This is an example of a karyotype. You can see chromosomes 1 through 22. These are our autosomal chromosomes, and then our last pair are the sex chromosomes. We put those in the back because, again, we can see the sex of the individual here. 1 through 22, chromosomes 1 through 22 are organized they used to be organized by people. Now computers do them. But this is a karyotype. And the reason why we go over this, before I get to the next part here, you can see the karyotype of a cancer cell. You can see what it looks like, those mutations, and what a cancer cell could look like, the karyotype of one. What can happen is you can have nondisjunction occur, which is the failure of chromosomes to separate during meiosis. And this results in sex cells having a chromosome gained or lost. So euploidy is the average number of chromosomes in a species. Aneuploidy is a change in that number. So what that nondisjunction looks like, so the aneuploidy is for this species would be 4, because there's 4 chromosomes here. This is normal meiosis. This chromosomes divide and split. We get N, which is the haploid number, which is 2. During nondisjunction, the chromosomes don't separate properly. And you can see we have some sex cells with an added chromosome and some sex cells with a missing chromosome. This can occur in meiosis 2 as well, but not all the cells then will be affected. So basically what this nondisjunction does is it makes sex cells with an abnormal number of chromosomes, either +1 or -1. Now, this is called monosomy or trisomy, and monosomy obviously is when they have one of the chromosomes in a homologous pair, so they're missing a chromosome. Trisomy is when they have 3 chromosomes, they have an extra chromosome for an homologous pair, and there's only 3 viable that beyond birth. All the other ones will be miscarried because having an extra chromosome will not be a viable option for a zygote to survive. So we need the right number of, I'll say right, but the 46 chromosomes in humans for it to develop correctly. So I'm going to show you some images here that are of individuals, and I just want to remind you that we are being respectful and just learning about these conditions as a scientist and as an educator and student. We have to be respectful of these people just going forward. All right, so trisomy 13 is the first one that we go over, and this is Pito Syndrome. This is individuals have major circulatory eye and brain defects, about 1 in 5,000 live births, and children, again, rarely live more than a few months because it's such a detrimental disease syndrome to have. Trisomy 18 is next. This is Edwards syndrome, about 1 in 10,000 live births. Almost all organs are affected. There is an especially small heart. And again, children very rarely live more than a few months. Trisomy 21 is Down syndrome. This is the most common form of the trisomy, and this is where individuals will have slight mental handicaps, short stature, flat face, heart problems. These individuals are this syndrome is the most common, 1 in 700 live births. These are the 3. All the other trisomies aren't viable in the sense of they won't be born. They're the trisomy, for whatever reason, won't make it through pregnancy, and there are only 3 that make it through pregnancy, and then 1 that can make it to adulthood, and that's trisomy 21. We go over very quickly. Again, I don't have you take notes on any of this. The couple types of trisomies and monosomies in the sex cells. So we have triple X, Turner's, Kleinfelters, and then we have one called the Super Y syndrome. Not this one. This one's not viable. You need an X chromosome. But first one's going to be Turner syndrome. You can see the X chromosome here. This is when an individual only has one X chromosome. They will develop as a female with slight irregularities, but 1 in 5,000 females. Next is Kleinfelter syndrome. You can see XXY. So this person has a trisomy and they have 3 chromosomes for their sex chromosomes. They develop as a male, but they have, again, physical irregularities. And 70% of the cases here go undiagnosed because these individuals will, the condition isn't as severe phenotypically, so they might not think something is wrong, which nothing is wrong, but they might not go to the doctors and look for something. And so these just go undiagnosed. Next is Super X syndrome. Develops as a female. You can see again slight physical irregularities. 90% of these cases go undiagnosed because, again, these aren't that severe where a person might think to go to the doctors. About 1 in 4,000 live births. XYY. This is our last one. This person develops as a male. Again, slight physical irregularities, and a lot of these go undiagnosed. So there is variation from the XX and XY chromosomes, and I like to go over these because it shows you that it's not just as simple as XX and XY. There is some variation.